always, and I always learn a lot. All right, so I will present here what we've been doing in my lab now at Barcelona Supercomputing Center over the past 3 years or so. I'm still in London as well, although not so much. And I just founded a startup called Graphlet Technologies, and I will talk about what components we are using for it. So just to motivate, we've seen many of you talk about different kinds of omics data. We're just learning more and more about less and less. We have all sorts of these things that you guys talked about, but also the foodome, the allergome, or whatever it's pronounced, and all other omes. So all of these data basically consist of molecular entities, or not even molecular entities, supramolecular entities, or cells, or tissues, or organs, or even entire humans. So they can be represented as nodes in networks. And then if there is some interaction or relationship or correlation between them, then an edge. So we have various kinds of, let's say, molecular networks that measure different things in a cell. We also have various networks where drugs could be molecules, and then, for instance, their chemical similarities could be edges, or if they cause the same side effects, it could be an edge or whatever. Then we have various kinds of ontologies. So we are all looking for the same thing. How much information can we, or knowledge can we extract out of these data types, sometimes in isolation from others, which is very important. But also what we care about in my lab is how to integrate them or fuse them together to see what collectively they tell us, and how to put that in a patient-centric way. And we design new methods, new algorithms, how to do this, because basically of the underlying computational intractability of many problems that deal with these large networks or graphs. So that's kind of in a nutshell. And now I will present just some illustrations of the methodologies that we've been developing. A couple of you have seen some of these, the first part of this previously, because it's from a paper from 2019. But others asked me to talk about it because it's not only the result of my ERC consolidator grant that's still ongoing, but also the main thing in my ERC proof of concept grant, with which we founded Graphlet Technologies. So this paper is about different types of omics data. We make them tissue specific for particular cancers and the control or proxies for healthy cells. Then we fuse them into this new model that we call an I-cell, or an integrated cell. So this is just another network. This is a new model of biology. And then we compare them. We compare them using graphlets. Many of you have heard me talk about it for the past 15 years or so. And then by this comparison, we get new cancer genes, and we got phenotypic validations of them. So let me slowly lead us through this paper. Now, these different kinds of omics data that we collect about the cell, they are qualitatively different. They measure different things. This is like putting different pair of glasses on to look at the same thing at the cell. And this is why when we look at the overlap in terms of the number of genes in the protein-protein interaction, genetic interaction, and co-expression, we have 3,000 roughly genes in the overlap, but only one shared interaction. And these are the databases that we used for this, and this is collectively for all of them. And there is no mistake here, it's just they measure different things. And because of almost no overlap here, this is why most of the other data integration or fusion methods fail. And when I say data integration or fusion, I don't mean amalgamation of the data, putting them on the top of each other, like everything together in a database, but I mean in terms of modeling, in terms of mathematics. So these are some of those that fail, and they fail for various reasons. Maybe they don't converge, or produce very large number of very small clusters, et cetera, et cetera. They all have different problems. And this is why we needed to come up with the new method. And this is how we went about it. So we created these tissue-specific networks. We get tissue-specific gene expression from the Human Protein Atlas. But keep in mind that this is not a microarray or RNA-seq. This is antibody staining, low throughput data, low number of samples. They don't represent all patients, just a small number of them. So later on, if you don't see your favorite gene that you love, then it was just not in the data. There's nothing wrong with the method, right? And then basically you overlay this on the top of these generic protein interaction networks, data from co-express DB or genetic interactions. These are static interactions to get these tissue-specific interaction networks. And also we only consider genes with expression available in the Human Protein Atlas and at least 2 actually protein-protein interactions so that these methods would work. So this is what I said. So nodes are genes expressed in the tissue from the Human Protein Atlas, whether it's cancer or control, and they're linked by interactions. And we took at first the 4 most prominent cancers in human and the control tissues of origin, so breast, prostate, lung, colon, rectal. And then this is just to show you that when we make these tissue-specific networks, both for control and cancer, they're pretty large data sets. You have on the order of 10,000 nodes, tens or hundreds of thousands of interactions. So basically you can use methods such as this one. Now this iCell prototype works as follows, and this is actually conceptually very simple. So you have these, so protein interaction network, co-expression, and genetic interaction network. So each one of them is genes to genes, right? In these matrices A1, A2, and A3, they're all color-coded. And then what you do is you approximate each one of them simultaneously as a product of 3 lower dimensional matrix factors, where you share this one corresponding to genes. These are genes here, and this is some low dimensionality, much lower than the number of genes. Let's say this would be, let's say 50, it's not 50, but let's say, right? And then, so basically this is the same corresponding to basically embeddings of genes in lower dimensional space that Patrick alluded to this morning earlier. And then these are specific to, basically these are encapsulated representations of these data. So how do you do this? You minimize this objective function basically, and then this converges to a local minimum. And then from this matrix factor, G, you multiply it by its G transpose, and then you get another G, genes times genes network. And this is our iCell. This is the integrated cell. And this is just an illustration of this iCell where we colored the genes by their functional annotation. And you see that in terms of reactant pathways, KEGG pathways, or go biological process, you have this just visually to see that it's functionally coherent. But not only that, actually additional functional information emerges from the fusion of all the data, despite of basically no network overlap. So how do you see this? When you make clusters, I mean, this is K-means equivalent. I mean, you guys certainly know about that. So then, and when you, in these clusters of genes, see how many genes are enriched, in this case, let's say in reactant pathways. And again, color-coded pink is for iCell, and the other colors are for here in the data, actual data from which we made the iCell. You see that in iCell, it's much higher enrichment, functional enrichment. So this is the new functional information that emerges from this mathematics, from this fusion, from this new model. And then you can test the contribution. So this is highly modular. I'll show you another example. So if you include all data, then basically you get the highest enrichments, for instance, here in Go biological process annotations in these clusters of genes. Now, let's go further. So we have these tissue-specific networks for cancer and control, these particular ones. So the 3 data types that I mentioned, PPI, genetic interactions, co-expression, but also their iCells. This is a new cell. This is the new, not cell, this is the new network. Now, in the Human Protein Atlas, a gene is either expressed or not in the tissue. So we find between cancer and control genes that are cancer silenced, cancer activated, always silenced, and always expressed. Now, this always expressed maybe at different levels. Let's revisit differential expression later. But what we find for now is only these always expressed genes are enriched in drivers. So not cancer silenced, not cancer activated, but always expressed, which is kind of interesting. This is why then we wanted to test the rewiring in cancer compared to control of these always expressed genes, and they're involved in cancer. So in cancer and control, iCell network, PPI network, GI network, and co-expression network, we find the most rewired ones, and now in terms of these graphlets. And I'll just put one slide about these graphlets because these were results mostly of my previous ERC starting grant that finished, oops, this is a typo here, it finished in 2018, basically, and as soon as after that, basically, I just started the consolidator that's ongoing. So they're still the best performing and most robust. It's a lively research area. If you put graphlet in Google Scholar, you'll get about 21,000 results. And graphlets are mentioned in about 300 patents. And they're used in many machine learning algorithms. They're generalized to work with directed network, with probabilities on the edges, on hypergraphs, basically that model multi-scale organization data such as protein complexes and pathways. We generalize Laplacians to graphlet Laplacians to take care of the expanded neighborhoods, not just direct neighbors. Then we generalize the spectral embedding and clustering as well. We just published, I think this year, graphlet Eichmann centralities, et cetera, et cetera. And we apply all that to biology, but others apply that to social sciences, et cetera. This is even image processing. It's a big research area. Okay, back from graphlets. Now we find the most rewired genes in cancer versus control in the iCell network, PPI, GI, and coax network, in each of them individually. And only in eye cells, rewiring is indicative of involvement in cancer, and the top 500 most rewired are enriched in cancer drivers and cancer-related pathways. So not in any of these rewiring, in PPI, GI, coax, no, only in eye cell, okay? So now to test this further, whether it makes any biological sense, we took top 20 of the most wired in cancer eye cells only, because in eye cells this works for the 4 cancers. So when you add 4 times 20, it's 80, but some of them repeated, so there is 63 unique. And we validated almost all of them either through literature, or we did our own knockdown experiments followed by cell viability. So you can see, for instance, if these genes, mutations of these genes in this cell line results in significantly reduced cell viability in cancer. And for instance, in this cell line, mutations of these in significantly increased cell viability in cancer. Also we went into survival, validated the others through retrospective analysis of patient survivals. And this is what I said I will say later, these differentially expressed genes. Out of these 63, only 17, so 27%, are differentially expressed in cancer. So the differential expression that we've heard about yesterday and today tells you something, but it's not the full picture. So each of the data types tells you something, but it's not the full picture. And you have to come up with these mathematical models to extract more out of all of these data collectively. So this is...
How do you define rewiring?
In terms of these graphlets, and I just put one slide. So basically, it's a methodology that we published a long time ago, I think 2007 in ALAT at TCCB conference. You were there, some of you were there. So basically, you take a look at the extended neighborhoods around the nodes. Maybe you've heard of network motifs. This is kind of like network motifs, so but there is a very important technical difference. So basically, you're looking like at these numbers of triangles, squares, paths, and stuff around one or the other, and how different they are in a nutshell. So there you have these feature vectors, counting these, and then you compare these feature vectors based on the counts of these small substructures. This is what
you're demanding a certain level of change in these motifs.
Yeah. Very big. Very big change in that. Exactly.
While we're at that, I was wondering something similar. When you say there was no rewiring in the protein interaction data, those are most of the time, reference maps that were collected in a single condition. So how did you assess, what was your measure?
This is the most rewired ones, right? So we took the most rewired ones in terms of graphlets, right?
Which data did you put in there? Yeah, but for the protein interaction data.
From I2D database, from Igor Yurishitsa's database from Toronto. He was my PhD advisor. So
right.
A big one, only experimentally validated data. That's the data in particular, for protein-protein interaction that we use here. So we found the most rewired in each one of these, okay? Checked in them enrichment in drivers, And only in eye cells, these top 500 most rewired were enriched in drivers.
I understand that, but I'm sorry for doing this, but
Yeah, so you have to give me 5 more minutes because I'm answering questions now.
For the protein interactions, there's very little cancer-specific data, right? So how do you?
Yeah. I think Patrick asked me that when we were at the conference you organized at Long Island called Spring Harbor. So, and I explained that before, so we have to go a few slides before. So it's here, how do I make them? So basically I take from the human protein atlas, these specific cancer-specific expression data and tissue control, and also in I2D, this is this protein interaction database, at least 2 PPIs, and then basically you overlay this on the top of this. Everybody does it in the same way. So this is how you get the tissue-specific interactions of whatever type, whether it's PPI or GR or whatever. But we can talk about it more if you're confused at the moment. Yes, so where were we? Yes, so, right, I think we were here. Then we went pan cancer. So once we found this for 4 cancers, then we added everything that was available at the time at TCGA, so 20 in total. So these are the cancers. We made their eye cells. And we're asking now whether similarly wired genes in different cancer eye cells are cancer related. We found just over 3,000 genes expressed in all cancer types. And then we found the most wiring similar out of them in these cancer eye cells. And then the top 500 were significantly enriched in drivers again, right? And then basically we found, so these are our top ranked ones based on this. And for instance, so basically we validated all of them nearly, but this one, for instance, we could not find any evidence, but then we went to Human Protein Atlas, we found that it significantly impacts patient survival in 8 different cancer types, and this is for pancreatic, this picture. All right, so, okay. Right, so basically this is about the eye cell. So differential expression or any other type of data carries limited information, and we can expand more if you fuse the data. This is also very generic. And then let me show you, so this was the first example for cancer, this eye cell paper. And let me show, this is unpublished work, we're just writing it up. So this is about rare form of thrombophilia. And with rare disease, you have very few genomes of these people. In this particular case, we had the somatic mutation of only 2 families, 2 brothers, 1 diseased, 1 healthy, and the daughter of the healthy brother that was diseased, who was diseased. And then we had 2 sisters, another family. So here are the genes and these these different kinds of molecular interaction networks that we've seen in the eye cell. So this is your eye cell. But here we kind of shyly add this one additional data type. These are your patients, these 5 guys, and then germline mutations. So here, by fusing this data in this simple way, basically you boost the signal. So because you don't have statistical power from the so few people, then if you add these molecular interaction networks, the thing is that there is a super exponential number of the ways this can be rewired. So you boost the signal by this data fusion, and you can find things out. For instance, this is a cluster of genes that in molecular networks that are characteristic to all diseased women. And this one is characteristic, these mutated genes, to the healthy brother. And this is probably what protects this guy from being diseased. So we are writing this up. Another one, so let me just now go into these different types of data. So this is your eye cell. This is, for instance, this one that we just saw, patients, 2 genes, let's say somatic mutation profiles, but you can add whatever you want in it. For instance, you can add these drugs, and then maybe chemical similarities are the edges between them, and then you have drug-target interactions here. And then you have these, that these drugs were given to these patients, or these patients could receive these particular drugs, etc. And then, so what we like about this kind of modeling is because it captures all systems level data, unprocessed, untransformed, at their full size, and also how the data relate. And this is the trick. If you capture precisely how the data relate, then you have very few parameters to tune. in this particular machine learning method. And not only that, it's explainable. So trivially, you get mechanistic explanations in the sense, let's say, I repurpose that this drug should be given to this patient, and then they ask you why. Well, and then you say, because this patient has these 2 mutated genes that this drug binds to. So this is kind of simplified. And what you're doing is then basically these matrices across the data types, you're simultaneously decomposing them into these lower dimensional matrix factors, by sharing the factors across the composition, you're ensuring data fusion. And all within one minimization function where you're adding prior knowledge or penalties from where these interactions are in the data types in your minimization process by these here traces of these here are Laplacians of your interaction networks, right? So now at PSB, now 6 years ago, we published this. This was from TCGA, serous ovarian cancer patients, 350-ish of them, where we used this minimization function and basically decompose these somatic mutation profiles and drug target interactions. And then from this one formula that you minimize once, you get patient stratification from the cluster of patients, you get cancer gene predictions from the clusters of genes that are enriched in cancer genes, and then you get drug repurposing from these drug target interactions, basically matrix completion property. And this again illustrates how modular it is. If you only use drug target interactions to predict drug target interactions, this is how accurate you are. But when you start adding more data, you're more and more accurate, and so forth. So this is 5-fold cross-validation, just kind of in a nutshell. And now the last 5 or 10 minutes I have, so we applied this to COVID. Very early on, we got the viral host interactions from the Krogan lab. We also got the differentially expressed genes in lungs. We wanted to bring the repurposing into the human genes rather the antivirals, which are tricky. So basically, we took the data from these databases. This is your 30-ish viral proteins to 300-ish human genes, viral host interactions. These are human molecular interaction networks. This is drug target. So drugs, 4,000 drugs to 2,000 something human genes, and then drug to drug chemical similarities. And then you decompose at the same time viral human matrix and human genes to drugs, sharing this factor to ensure the fusion, and then you get co-clustering and matrix completion for drug repurposing. Once we repurpose the drugs, then we were wondering, because there were quite a number, like which ones should we repurpose? The one that hit receptors, everybody loves these drugs that bind to receptors, or where do you hit? And then we took these human viral interactors, basically. So, and then we took their neighbors, and then we took differentially expressed genes, like should we hit differentially expressed genes? Everybody loves differentially expressed genes. And then the neighbors of differentially expressed genes and the neighbors of these viral interacting proteins. And it turns out that only these common neighbors are enriched in viral functions. And these are enrichments, some of the enrichments. Nothing else is enriched. Differentially expressed genes are not enriched in viral processes, nor these receptors, etc. And these are very unique, these common neighbors, as we call them, occupy very interesting topological part of the network, very central in terms of many measures, including graphlets. And then when we saw, so basically there were around 50 genes there and targeted by around 150 drugs. And out of these 150 drugs, around 30% already, were either in Gov trials, database or in Coordate database. So these are validations that we predicted quite correctly, 30%. And then at the time I had a biology postdoc who since moved to industry somewhere in Germany, went back home when Corona hit. And then, so basically he did some analysis, and basically all of these converged to VEGF and nitric oxide signaling, but I don't have the time to go into this. And these are some of these top predictors. Just to say that with these methodologies, we are analyzing single cell expression data, fusing them with networks. This is with Alex Cooping as part of the ITN project. This is Marie Curie project. So what they do in their lab, they start from pluripotent stem cells with and without a mutation that's for Parkinson's important to them. And then they measure with single cell draw-seq analysis at day 0, day 6, day 15, and day 21 of controls and Parkinson's disease. Basically, they measure this expression. So this is the number of genes that were measured, the number of single cells. And then basically we are fusing this data with 4 different types of networks, or 3 types of, no, 1, 2, 3, 4 types of molecular networks. These are single cells to genes. This is expression of these genes. And this is again data fusion. And we are finding basically where and when this change happens and how to intervene with already known drugs, what we could perhaps repurpose. And in the end, in the last 2 minutes, so basically this is one of the papers from last year by a very talented PhD student of mine, Alexandra Zenos. So we are using the similar thing to do gene embeddings. We've seen some, so basically network embeddings. We've seen some that Patrick mentioned this morning. And basically everything that you've seen, you can consider it as an embedding. But let's say in this particular paper, protein interaction network, this is human protein interaction network. By this methodology, we're embedding in lower dimensional space where we can control for orthonormality, so to minimize the dependencies between these bases of the space. Then basically we are seeing what's happening in the space. So in the space, the genes basically that were clusters in the network, we would like them to come close in space, and they do. And we find that the space is functionally organized. Basically, what comes close to space, basically these genes belong to the same functions. So just in a nutshell, then what is very interesting here is, so you have, for each gene, you have a vector in the space. And now you can do simple linear operations with these vectors, such as you add them and you have to find the average. And basically then you find the proximity to those points with cosine, and then you can predict new members of protein complexes and also cancer genes. And we found some, these are some of our predicted cancer genes, and we validated all of them. So that's kind of that. So just to say that this is a very versatile machine learning methodology. You can put in it whatever you want, like EQTLs that we listened to about just at the talk before, or somatic mutation profiles, expressions, methylations, copy number variations, clinical profiles, similarity, treatments, adverse effects, whatever you want. It's here. However, each time you pose it, it's different NP hard, continuous optimization problems. You have to propose objective function optimization solver, prove convergence, correctness, and optimization is slow. And this is why we are at the Supercomputing Center and we use it extensively. So, but maybe with these embeddings, we can do better. So now to conclude, we have seen the importance of precision medicine and the strategic intelligence of the World Economic Forum underlined. There's artificial intelligence and data science and multi-omics science here, and et cetera, et cetera. It's all around there. I hope I conveyed the message that we need to merge them all together. This is a result of my ERC Consolidator grant, with which we make new AI methods. And also we are commercializing it. We just founded this graphlet technology startup. We are doing de novo drug discovery, precision drugs in silico, that is disease and target agnostic. And we are also, in addition to that, doing many other tasks of precision medicine from all omic data collected, kind of using what you have just seen. And not to forget to train new people. Everybody's going to industry these days. There's going to be nobody left in computer science to teach the people anymore. So we published this book a couple of years ago at Cambridge University Press for basically training embedding data scientists who would like to do this multidisciplinary work. as a textbook for PhD students and postdocs. And in the end, for funding, I thank the organizations that supported me over the years. Of course, my group members, without whom none of this would be possible. This is part of my group in Barcelona. Past members, collaborators, and learned societies. And I'd like to thank you for your attention. Yes, there is a.
So obviously, since I'm not sure data in many different ways, including your analysis, one coming from last group and some others as well. So just wondering at the end of the day, what is the overlap in the predicted drugs? Have you been able to look at it? Because it feels that the data may be related. You would hope that how much of it is data, how much of it is method, do those repurposing effort comes with a similar have any insight on that?
I don't know. I don't remember if we did. This was a very early study, and now we just submitted it these days. Or no, we asked to submit the next one, in which basically we compare to this one and last one and stuff. But you know, once your paper is in and his is not out, you cannot compare. It would be interesting, but honestly, to me, I mean, does it work or does it not work? If his method finds something else that works and mine something else, it's cool. I mean, this is all anti-hard. I mean, so these are all approximations. So let's find something that works. This is my approach. But this is what you're asking. It's kind of more theoretical question, you know, and I'm not sure if we can. Okay, I think we could get an answer to that, and it would be about exploring the properties of the data and the properties of the question you're asking. And then maybe you can come up with some general rules, and we have some ideas about that in general. Yeah.
One of the methods are linear algebra, basically. So do you have to pay some attention when you combine all these very different data types that you can actually do linear operations to combine them, or is it trivial that you can do this?
Well, in the light of what I just showed you in the end, and so this is just the first of the series of papers. We have another one submitted, and I think 2 or 3 more on the roll being written up. It turns out that the space is linear, and there are reasons why it's linear and which kind of data produce this linearity in the embedding space, and this is why this works. But let me just say that, and then when the paper is published, then it will be. Sorry.
Oh, so let's thank Natasa again. Reminder, don't yet, escape. We have a good photo now at the front, and then you are free for lunch, and we meet again at